So we did talked about metabolism being composed of anabolism and metabolism, and we're going to see examples of probably only one of those today. Later on in the semester, we'll see examples of a couple of them. Actually, we'll see both today. Okay, so what we're going to do before we actually get into all of cellular respiration is we're going to talk a little bit about enzyme functions. And this first part of this stuff you will have heard before, but I'm actually trying to concentrate on an aspect of it which will apply directly to human physiology. So even if you've heard the terms before, please listen to the spin, listen to where I'm trying to go with it. So before we can get into metabolic pathways like cellular respiration, we need to talk just a little bit about enzymes and make sure we have sort of a common experience with what enzymes do. So enzymes and enzyme functions. So this is the way it was always taught to me in chemistry, is that enzymes lower the activation energy or catalyze a chemical reaction. The problem with that for me, and maybe not for you, is that I got really confused when they were talking about lowering the activation energy, and then, for instance, they would tell me it's an exergonic reaction. I'm like, well, you just told me it didn't require energy, and now you tell me that you lower the energy required for the enzyme or for the chemical reaction. So there is another way to think about enzymes, which is true and makes more sense to me, and also dovetails really well into human physiology. So it does lower the activation energy, but what it does for a chemical reaction is makes each chemical reaction statistically more likely, and therefore the product will build up more quickly. And so I'll show you sort of how this works. So you guys, of course, know that enzymes are proteins, and proteins are very shape-specific in their interaction, right? Everybody knows that. Okay, so what I'm going to do is sort of give you a fake chemical reaction that I want to occur, and an enzyme to catalyze it. So I'm going to take A plus B, and I want to make this product that is C. Okay. So before we go any further, some terminology that you've all known for a long time. If you have a unidirectional chemical reaction, then of course these are the reactants, and these are the products. But if it's an enzyme-catalyzed chemical reaction, the reactants can also be considered substrates in that they actually fit into the enzyme. So let me give you a visual to make that make sense to you. So if we say that this is enzyme number 1, shape-specific, and it has a shape-specific binding site for A and B, you can see that in the presence of the enzyme, you will end up with a lot more C, right? Does that make sense? So if this were an intracellular chemical reaction, and I was just waiting for A and B to smash into one another to form C, I would probably get some C, right? But I would get such low levels that if I needed that product to support homeostasis, I would be a dead cell, right? But if I want to make it statistically more likely to get product C to form, I can have sites where A and B are both attracted, right? And so the way that makes sense to me to think about enzymes is just to think about them with statistical probabilities, right? If they're more likely to encounter one another, you're more likely to get a product, and therefore you're going to have more product over a given period of time. Does that make sense? It is also true. It's not generally the way that it's approached, but it is also true about chemical reactions. So let's make sure we've got all the players down. So you know that the reactants are now called substrates. Substrates specifically will bind to the active site of the enzyme, or sites of the enzymes. And that's sort of the definition of the active site that binds the substrates. Okay? Nothing else can bind them. There may be other binding sites, but they are not going to be active sites. Okay, keep that in mind for a second. And then, of course, the thing that you have heard a million times before is that the enzyme is not destroyed or consumed during the process of the chemical reaction. You can reuse it. Can you reuse it infinitely? No. But let's just look at this basic visual that you're seeing. This enzyme will fit the substrate, right? But then the enzyme substrate will actually change the shape of the substrate, right? When the substrate changes shape, it no longer fits the site, and then it falls off as a product, right? So you're just dealing with shape interactions. And then notice that I can reuse this thing. Can I reuse it infinitely? No, because you can wear it out like every other biological or chemical process. So it's not as if you get to make your enzymes once during infancy and they last you your entire life. But you can use them repeatedly. Like you can use your washing machine over and over to wash clothes, and stick clothes in there, but you're not going to have the same one that you had when you were 12, right? It ain't going to last that long. You're going to be able to, eventually you're going to have to replace it, okay? So you can reuse these and they're not changed permanently, but they will actually wear out. Okay, so now what I want to think about is try to get you guys to predict, and you'll be pretty successful. Most of these are just little logic ideas. What can I do that will impact the rate at which C accumulates, or the rate of an enzyme catalyzed chemical reaction, okay? Some of them are really easy to predict, like for instance, would I get more C if I added more A and B and still only had one enzyme? Yeah. No, right? You're not going to, if you only have one enzyme, it doesn't matter how much substrate you dump in, right? It still has a limited rate at which you're going to accumulate C. Likewise, would I get appreciably more C if I added a whole bunch of enzyme and kept the the substrate concentration very low? No. Okay, so conceptually those things are going to make sense. Let's just look at them visually, and then there's going to be some more that are going to make. Okay, so the enzyme activity and therefore the rate of the chemical reactions can be affected by, one of them is called saturation. I know you've heard this word before. Okay, so please look at what we're saying. We're saying as substrate concentration increases, the chemical reaction rate will increase until what occurs? Saturation. Now, what is saturation? What is physically happening?
There's a whole bunch. So there's not enough enzyme.
There's not enough enzyme. And so what's happening is your enzymes get full, right? And if your enzymes are full, right, then everything's just waiting in line, right? You can't increase it any further unless you do what?
Increase one.
Unless you add more enzyme. Does that make sense? So when you're here, I could continue to add as much substrate as I wanted, and I would still have a limitation on the rate of the chemical reaction because of the amount of enzyme that was present, right? And that's called your saturation rate. Conceptually, the only way that you can increase your saturation rate is to increase the amount of enzyme that you have, right? Is that an instantaneous cellular fix? No, it is not. Protein synthesis is not instantaneous. It actually takes a little while. And by the way, this is not usually a good solution, a good short-term solution, but you can run into situations in which, okay, remember how we were talking about like the error message that you can get? So, an adaptation and accommodation. So you move to Utah and all of a sudden you can't walk up the stairs, right? And you've got chronically low levels of oxygen, and you have that sort of consistent error message that you are not carrying enough oxygen. Can you see that if you get that over a little while, you might go, oh, I actually need more enzymes involved in hematopoiesis or red blood cell production, right? So it's more of a long-term adaptation or accommodation fix that you could make. You could change a set point, but you don't change a set point every time you get a single error message. So protein synthesis is not instantaneous. It's also very expensive. So you can change your saturation rate, but it doesn't happen as frequently as you imagine. So you can change the level of enzymes. So the only way to really change the saturation rate is to impact the enzyme concentration right is here. So enzyme concentration, you guys know, but tell me, as enzyme concentration increases, reaction rate tends to do what? Increase, exactly. And you do that by basically changing the saturation rate. Okay, so now let's go back to the whole concentration of substrates. The substrate concentration, so this original graph that we looked at said substrate concentration, as substrate concentration increases, reaction rate tends to do what? Increase until what? Until saturation, exactly. So it does tend to increase with an asterisk, right? Until you hit saturation, and then it's basically capped. But notice it doesn't go back down to zero. It just like stays, they're all sort of waiting in line for the next enzyme. That's what they're doing. Okay, so now these two are going to be related to one another, so I really want you to listen to the difference between them. Okay, so now we have this concept of substrate specificity. And substrate specificity says that, so they usually teach you that an enzyme only fits one substrate, but that's really just kind of a useful lie. Because there are so many very similarly shaped molecules in your body, like some amino acids are just like a single bond away from one another. So what happens is an enzyme has a favorite substrate. This is his best buddy right here. But in a pinch, in the lack of C, maybe A will do, right? Never, ever, ever will B do, right? And maybe, you know, when times have been long and lean, you could actually fall for C, right? So the deal is that there are substrate specificity. There are substrates that actually bind very well and very readily to the enzyme, okay? And there are substrates that don't bind as well. So what do you guys think? As substrate specificity increases, what can you predict would happen to the rate of chemical reaction? It's going to go up, exactly, because you fit quickly, right? As you fit quickly, as substrate specificity goes up, the chemical reaction rate would be expected to go up.
You mean that when a substrate becomes more specific about which?
No, when I use this one instead of that one, that's what I'm saying. It fits better. So this concept implies no, the substrate better fits the enzyme, okay? So the concept here is that there are multiple possible substrates, and the one that fits best gets the fastest rate of chemical reaction, right? So it would go to saturation fastest, right? And it would also be fastest globally because it would accomplish the product, fall off, you get another one, accomplish the product, fall off. Does that make sense? Okay, so the concept of substrate specificity, just listen to this and keep it in mind for when we do the next one, implies multiple possible substrates. Okay, one enzyme holding the enzyme constant, multiple possible substrates exist. Okay, so keep that in mind and now contrast it with this next one. And the next one is called enzyme substrate affinity. And the enzyme substrate affinity concept says that, okay, so now what I'm going to do is I'm going to compare us two, okay? I'm going to give both of us the same substrate, okay? But are her enzymes identical to mine? No, they're similar, but they're not identical. Do you think that there are enzymes which fit the substrates better and worse, holding the substrates constant? Yes, right? That explains a lot of the metabolic differences between humans, right? So if I had another woman who's my age, my approximate size up here, dump substrates on both of us, identical substrates, would we respond identically? No, we wouldn't, because our enzymes vary, okay? So with enzyme substrate affinity, what does the word affinity mean? Attraction, right. I have an affinity for golden retrievers and champagne and red shoes. Yeah, red shoes, I do. It basically means how well the enzyme and a given substrate will actually hold to one another or be attracted to one another. So the difference between substrate specificity and enzyme substrate affinity is substrate specificity implies multiple possible substrates holding the enzyme constant. You guys with me? And so enzyme substrate affinity holds the substrates constant and acknowledges that there are multiple possible enzymes. That makes sense? Okay. So are there slight variations in enzyme shape that might make it fit better or worse? Exactly, right? So we will have that obviously between people, but also within me, since I have two parents, I very frequently have two copies of the same enzyme, one of which has a greater affinity for the substrate, okay? So the concept here is that if you have an enzyme with low affinity for the substrate, the chemical reaction is going to be much slower on the upsweep, slower to get to saturation. If it has a higher affinity, you're going to get there more quickly. Does that make sense? Okay, so I'm going to back off for just a second. I'm going to ask one of you guys to tell me in your words the difference between substrate specificity and enzyme substrate affinity. And I'll be nice to you, I promise. When I ask you to do this kind of stuff, I'm never a bastard. Well, I don't think I am. Yeah. It's all very subjective. No? Want to try?
So the affinity is the attraction to the enzyme. Right?
Okay, yes.
And then the other, what was the other one you said?
Specificity.
So the specificity is how well it fits into the enzyme.
Yeah, those sound very similar though. You're going to have to like separate them out just a little bit to make sure that you would be able to tell the difference on a test question. So what is the real big difference between them? I know, I just said it just a minute ago. Can you do one of them? Yes. Which one holds the enzyme constant and varies the substrate? Substrate specificity. Which one holds the substrate constant and varies the enzyme? enzyme substrate affinity. So do you see that that's the nugget? That's the difference between the two of them, okay? I have no idea what I just said.
Which one holds the
Substrate specificity holds the enzyme constant and varies the substrate. Okay? And any good scientist knows that you only vary one variable at a time, right? And the other one, substrate specificity, holds the enzyme constant and varies the substrate. Enzyme substrate affinity holds the substrate constant and varies the enzyme. So if you notice, the thing that varies is in the title of each one. Do you see that? Yeah? Okay, go ahead.
So if each enzyme is accepting a substrate that's like its second or third choice,
Yeah.
will it affect anything other than the rate? Like, does it ever get a product that's...
Yeah, you can get a product that's inappropriate.
I wondered if it was like a 0 or 1 thing. Like if it's
It's not actually. A lot of times when they do fit something, it's like, that'll do as far as an intermediate product. Not always, but we're going to talk about biochemical pathways next. If you had the inappropriate thing binding, it might actually impact the entire biochemical pathway. Although you can see how that would be selected against, right? If you gave yourself 10,000 years of evolution and that never got you the proper product, I'm pretty sure you wouldn't be the one getting to have sex and babies, right? You'd be selected again. So it depends on what biochemical pathway you're talking about. Okay, so you'll get comfortable with that. Just give yourself a minute to think about it and separate it. And then, so the only other thing that we need to talk about, because we did saturation, we did substrate specificity, substrate concentration, that one, enzyme concentration. Okay, so then the last one is probably the one that you haven't seen before, or at least I hadn't seen it before, physiology. And it is looking at what the product concentration will do to the rate of the enzyme catalyzed chemical reaction. So in order to get you to understand that, I'm going to make a hypothetical biochemical pathway. And you know, biochemical pathways is just a fancy way of saying a series of enzyme catalyzed chemical reaction. Because inside the body, you usually don't get your product from a single chemical reaction. It might be 20 of them in series, right? So we're going to look at a fake biochemical pathway, and then after this, we're going to look at a real one. So here's what I'm going to have. I'm going to start out the same way. I'm going to start with A plus B goes to C. But C isn't the product that I'm interested in. I'm interested in F, okay? This is catalyzed by enzyme number 1, but C is going to dissociate to D plus E, and that's catalyzed by enzyme number 2. And then D plus E is going to be catalyzed by enzyme number 3 to F, which is the product that I'm interested in. Okay, so what I'm going to try to figure out is what the concentration of F will do to this chemical reaction. So let's look at it visually and see what's going on. Okay, so we're going to start out the same way. We have A and B, which are that shape and bond here to the active site. Okay. That's how we started. However, we do have another binding site on the enzyme. It's not the active site. It can't be. Otherwise, this won't work. It's another binding site up here, let's say, and we're going to call it the regulatory site. Good? Okay, so now I'm going to tell you to look. Lo and behold, F is a delightful fit at the regulatory site. And now I want you to see if you can predict. What do you think F binding to the regulatory site will do to the shape of the active sites? Should it make them fit better or worse? Worse. Exactly. Do you see what we've got here? It's an example of what kind of feedback? Yeah, it's an example of negative feedback, right? Do you see that? As you start to accumulate more of the product, then you end up inhibiting the actual first step of the enzyme catalyzed biochemical pathway. So a logic question for you. Why not have it feed back to E 2, 3, 27? Why is that less common than feeding back to the beginning of the biochemical pathway?
It's a cascade effect, right?
It's a cascade effect, and what would happen if you were making all of those intermediate products? Yeah, you're wasting materials, right? And also, what if some of them were energy requiring reactions? You're wasting ATP. So it's very likely to feed back to the beginning. Sometimes it's the beginning and a couple other places because it wants to make sure it stops, but it's almost invariably back to close to the beginning. Does that make sense to you guys? Okay, so if you looked at that and it really worked this way, and the product concentration tended to decrease the rate of chemical reaction ultimately, then what would happen if I looked over time at the concentration of F? What would it do over time? What would it do? So as it went up? Yes, exactly. What does that look like to you? Homeostasis, actually. This is a cellular example of negative feedback to accomplish homeostasis. This is a classic homeostasis example, right? So as long as, do you see that this is just statistics and probability? The more F you generate, the more likely it is to find enzyme number 1, right? And as you turn off enzyme number 1, then less F just gets generated, right? And then less of those are full, right? And so you tend to oscillate over time about an ideal range for the concentration of whatever product you've got. Right? Go ahead.
I just wanted to go back to enzyme concentration for a
Mm-hmm.
second. So can enzyme concentration be the same as with the substrate concentration where the reaction rate increases with the asterisk that it's with enough substrate?
Oh yeah, you could put that substrate. Yeah, you could put that in there for sure. The way that your textbook represents it, they do provide enough substrate, like the substrate concentration continues to go up. But yes, that's true too. Okay, so is everybody okay with the whole product concentration idea? So this is a classic example of negative feedback. You may have never seen it at the molecular level before, but now you are seeing it at the molecular level. Okay, so what we just saw is something that you probably knew before, but if you didn't, that enzymes can be turned on and off, up and down. And so conceptually, probably will make sense to you that you can make a whole bunch of enzymes in a cell, but do you want all of them on at all times? No. Do you want to be able to turn them on and off? Why don't you just make them when you need them? Because there's a tremendous lag time in basically how the cell is going to respond. So conceptually, you need to be able to make the enzymes, and some of them will be default off because I don't use them very frequently and I can just turn them on when I need them. Others I use almost all the time, right? But sometimes I need to turn them off. So they will be default on and you can turn them off. So there are abilities to turn on and off enzymes so that you can change the cellular activity. And so this is one of the ways that you do it. This is called allosteric regulation. Allosteric implies shape. And so what happens here is just like this example. So here's an enzyme with an active site, right? And I have a modulator. A lot of times they call it a ligand. It's just a general term for something that binds loosely and reversibly to not the active site, but the regulatory site, okay? If it bound to the active site, it would be one of the substrates and not actually a modulator. So what's going to happen is the modulator binds to the regulatory site, and notice what it does. It temporarily changes the site of changes the shape of the active site. Okay, so you could turn it on, you could turn it off, it doesn't matter. What matters is that it's loose and reversible. Generally, this type of interaction right here is usually just a polar interaction, like electrically attracted, or a hydrogen bond. Sometimes it's an ionic bond, okay, but a loose, relatively reversible. And so what's going to happen here is you can see that this is without any activator at all, okay. This is when you put in the activator, right? This is one that needs to be activated. If you put in the activator, it'll actually get to saturation more quickly. And this is one that is actually going to be inhibited. So look here, okay? This is how general enzymes look. They're going to get up to the saturation rate at a pretty much linear. But here, you actually are going to need to be activated to get up to the saturation rate. So this is an allosteric enzyme. This is a classic example of allosteric regulation right there. It's a loose reversible interaction, turning the enzyme on and off. So with allosteric, it has to be able to fall off by itself, right? You attach relatively loosely, you fall off by yourself. So what is an example of this in the human body? In the human body, well, this is an example. We'll run into more, but enzymes often require what we call cofactors or coenzymes in order to fit their substrates. And I bet you've heard the word coenzyme, especially if you're like into health food. They like to sell coenzyme Q10. Some of it's BS, and some of it actually helps a little bit. But what the hell are they talking about when they say it's a cofactor or a coenzyme? It is something generally needed in trace quantities that binds to the enzyme and usually makes it fit the substrate better, okay? So that your chemical reaction is more likely to occur. In you, the most common things to behave as modulators are vitamins and minerals. So let me put this together for you. Generally, at this level of your education, you're going to know that you need vitamins and minerals, but you also probably understand that zinc doesn't end up as part of your DNA structure, right? You know you need it. You also know that it doesn't actually end up as a structural component of your large organic molecules. So how the hell are you using those things? The way that you are using vitamins and minerals that you need in trace quantities is that usually they are cofactors or regulators for enzymes that are part of that metabolic process. So if, for instance, you were iron deficient, you could see a tremendous impact on your metabolism, right? You added it back. Was it directly involved in the metabolism of most things? Red blood cells, yes, but most things not. Okay, so they end up basically being the rate-limiting factors that actually make your substrates fit your enzymes better. Okay, vitamins and minerals do that. So the difference, by the way, between a vitamin and a mineral, they're both needed in trace quantities. They are not catabolically broken down. They're needed in trace quantities, but vitamins are organic. They're generally carbon-containing and came from living things. Minerals are inorganic, and you could get them by licking a rock if you could just find the right rock, right? You can lick calcium off of a rock if you want to. So that's what vitamins and minerals are. Fascinatingly, one of the other cool things about cofactors or coenzyme is sometimes they use the cofactor or coenzyme, which you know is going to be needed 26 steps down the biochemical pathway, and is sort of the rate-limiting factor way down there. So they'll use the coenzyme, the modulator, the coenzyme, as an indicator that it's safe to start the process for enzyme number 1 if I know I'm going to need it in very small quantities for step number 27. Does that kind of make sense? Little check mechanism. Do you have it? Then you can start going, and if you don't have it, then you might as well wait until it accumulates, and then you can go. So that's cofactors and coenzymes and their relationship to vitamins and minerals. Okay, there is one other way of doing the same thing, of changing the shape of the active site, and it's called covalent regulation. But look, notice what happens to the active site in this picture. Changes, right? You're going to accomplish the same thing in the next figure, right? Active site changes, but you're going to accomplish it by a different mechanism. This one is not as reversible, it's more likely to be expensive, and it's a little slower. So with this one, it's called covalent regulation. And this time, instead of using loose interactions like electrical interactions or hydrogen bonding or ionic bonding, I'm going to use a covalent bond. And covalent bonding changes the activity of the enzyme. So what I'm going to do is I'm going to need friends in order to do this. I'm going to need an enzyme that will help me to add a covalent bond and an enzyme which can break it. So the way that you might have seen this example before is with phosphorylation. Phosphorylation is a really common way to turn things on and off. Like you pop a phosphate off of, what usually do you get your phosphate from? Yeah, you usually pop it off of ATP and then slap it onto something else, and that will change the activity of that something, right? And then in order to turn it off again, you yank the phosphate off, usually give it back to ATP, right? So I know that you have probably heard these words before, but you are going to hear them later in Phys 2. A kinase is something that phosphorylates an enzyme and therefore changes its activity, okay? So here, in this instance, a kinase would actually phosphorylate the enzyme right here. So which, enzyme A or enzyme B? And if this were dealing with phosphate, which would be the phosphate, enzyme A or enzyme B? A, yes, because it's phosphorylating, right? Do you see it? It's adding that inorganic phosphate. And then this one right here is going to be the phosphatase, which pops it back off, okay? So this is not as reversible, it's not as quick, it's more likely to require energy, but it's also more sustained, right? If I wanted it on, stay on, damn it, until I tell you to turn off, then this is a good way to accomplish it, right? The other one's loose and reversible and really impacted by product concentration and that kind of stuff. Does that make sense to you guys? Okay, so by the way, these are not supposed to represent chemical reactions. It's just an abbreviation that shows you that what a kinase does is takes an enzyme, adds an inorganic phosphate, and then you're phosphorylated. And the phosphatase does the opposite, right? Takes a phosphorylated enzyme and pops the phosphate off and gives you an inorganic phosphate. Does that make sense? Did you have a question? Did you have a question?
I did, but you answered it.
Okay, all right, so you got it? So, but notice that basically what you're doing is still just changing the shape of the active site. You're changing it temporarily, and you can change it back. Okay, all right, so with that in mind, now we are going to get to metabolic pathways. And so a metabolic pathway, this is a fake metabolic pathway, right? It's a series of enzyme catalyzed reactions to ultimately get me the product that I want. Real metabolic pathways are probably the reason that some of you guys hate chemistry, but we're not going to do them quite that way, but I will show you some real metabolic pathways. This is the metabolic pathway that is the first step of cellular respiration, called glycolysis. It's a series of 10 enzyme catalyzed reactions. No, you do not learn it in this detail in here. In biochemistry, you will. Hexokinase, phosphoglucoisomerase, at each step, there is a different enzyme involved. Okay, so this is a biochemical pathway. It's the first step of cellular respiration, but what we are going to do is take cellular respiration and make sure, even though you've learned it before, that this time you get to the point that you can discuss it in the context of human physiology. So that's my goal for you. Not to teach you cellular respiration for the first time, but to teach you it so that it's applicable to what we're going to learn the rest of the semester. Okay, so one important metabolic pathway breaks down organic molecules to ATP, and that is a what? Anabolic or catabolic process? Catabolic process. And so what we are going to do is to try to generate ATP. This process is called cellular respiration, arguably the most important one for you. But if you were a plant, you could actually do photosynthesis too, and then you would be even cooler, right? Plants make the sugar, use the sugar, make the sugar, use the sugar. So as cool as you are, you'd be cooler if you were a plant. I don't even like plants. Like when we did molecular biology, I could not remember any of the plant stuff, but they're really awesome. Okay, so that's only the first step of cellular respiration. Here's the second step of cellular respiration, which is Krebs, okay? And here is the third step of cellular respiration. There's a third step of cellular respiration. That's not how we're going to approach it, though. That's a way that tends to make people's eyes glaze over, make them think they want to change their major. We're going to approach it. First thing I'm going to try to do is what I had to do for myself to ever remember anything about cellular respiration, and that is to make it relevant, to make you give a crap about something that people have tried to teach you 2 or 3 times. And if you were anything like me, you learned the minimum and then dumped it as soon as you possibly could. Hopefully some of you guys are better students than I was, but I just couldn't see how it was relevant to anything. So the first thing I'm going to do is try to teach you how relevant it is and how you will never escape it for the entire rest of the semester. So you might as well take a bite, chew. Okay, so we're going to start with a simplified version, a unbalanced summary equation of cellular respiration, okay? And specifically, we are going to do cellular respiration of glucose. We will learn, of course, that you can burn other sugars, you can also burn fats, and you can also burn proteins. But they always start with the classic, in part because this is your brain's favorite, okay? So the unbalanced summary equation takes glucose and oxygen gas and generates through a series of about 40 chemical reactions, generates carbon dioxide, water that we call metabolic water, and the big picture is this. Okay, so how does this relate to what we did in the last set of notes? How is ATP related to homeostasis? ATP is the thing that keeps you from looking like a flesh-colored pancake, because it is actually ATP that battles entropy, that allows you to not become one with your external environment, because that, for a human, is death, right? So we need to constantly sort of battle against becoming one with the external environment. This is your source that maintains homeostasis, okay? So this is how it's related to the last stuff that we did. Okay, so what we're going to do is look here. This is the entire reason, by the way, and how it's related to homeostasis and physiology is this equation, from my perspective, is the foundation of the reasons you eat, breathe, and have a cardiovascular system. You eat because you need this or something like it, right? You breathe because if you had this and not that, you would get very little ATP, not enough to maintain homeostasis, which we will actually see. So you breathe because in order to get the most out of your nutrient molecules, you're going to need oxygen to break them down. You also breathe because you need to get rid of this. Can somebody remind me one of the impacts of accumulating carbon dioxide?
Acidification.
Yes, causes acidification, which will, of course, challenge your homeostasis, right? Change the shape of your proteins that we were talking about. Water is not much of a problem as far as a byproduct. This is metabolic water, but it just mixes with all of the other water in your body, ain't no big deal. And then ATP is, of course, the thing that helps you to maintain homeostasis, to battle against the external temperatures, changing your internal temperatures, to replace your blood pressure when it gets too high or too low. It's ATP that allows you to do that. Okay, so this is the entire reason that you eat and breathe, and the reason you have a cardiovascular system is because this doesn't diffuse quickly enough to keep you alive. So you are going to have to circulate. Okay, all right, so now what we're going to do is we're going to go through this 3 stages of cellular respiration. We're going to simplify them, but we're going to try to make them meaningful. My objective when we finish this is that if you really understand how cellular respiration applies to human physiology, then you could teach it to a smart 7-year-old or your grandma who'd never heard it before, right? Because you know that when you understand a complex subject well enough, you can teach it simply, correct? So that's what we're going for here. We're going for the point that you can teach it simply. Okay, so cellular respiration, the most important metabolic pathway in you, starts with glycolysis. And so I will remind you what glycolysis looks like, and then simplify it dramatically, because we are not going to learn those 10 stages. We are going to look at it as a big picture overview like this. Okay, so here's glycolysis. What happens in glycolysis is glucose and other simple sugars will go into glycolysis. What's going to come out the other end is called pyruvate or pyruvic acid. Glucose is a 6-carbon sugar, right? Pyruvate, you get 2 of them, and they're each 3 carbon molecules. Okay, everybody okay? So notice that the name implies what it's doing. Glyco, as in sugar, right? And lysis, as in break, exactly. So you probably won't be surprised that almost anything that is not a sugar skips glycolysis if you're going to burn it. Just briefly, look at where proteins go into all of this after glycolysis. Look at where fats go in after glycolysis, except for this one, which is glycerol, which is kind of a sugary fat. That one goes in there. Go ahead.
It's kind of off topic, but you said that it's the brain's like primary way to use things. Why do they use glycolysis if like fats generate are like more efficient, like 9 grams per?
Why does the brain use glucose?
Or like why have we evolved to use glycolysis rather than like fat? Because fat's like what 9 grams per whatever.
You know what? I haven't thought about that before, but I have the seed of an idea. Let me work that around in my head and see if I can come up. I mean, you know, we never know the answer to why we evolved this way. But sometimes there is a backwards reasoning that can make it make sense.
It can run on that stuff.
What?
It can do it though, the ketogenic diet.
Yeah, the brain has a very hard time. What the brain actually does is takes proteins and makes them into glucose and then burns glucose. Or, right? So, so that's definitely going around your ass to get to your elbow as far as like getting the brain running. That's how we say it in Texas. Do you guys not say that here? Okay. I always forget that I'm in Utah, right? That's like effective teaching tools when you're in Texas. Go around your ass to get to your elbow. Okay, so I'm going to think about that one, though. I think it's a good question, and I have the seed of a thought about it, but I don't know the answer. I'll hypothesize the answer with you at some point in the future, though. Okay, so let's talk about glycolysis. Glycolysis is the first step. And then we're going to run into the Krebs cycle, which is in the middle, and then we're going to run into oxidative phosphorylation, also called electron transport, okay? But let's talk about glycolysis in a big picture. So glycolysis is a series of 10 enzyme catalyzed reactions. We're not going to memorize the 10. We're going to memorize what goes into glycolysis and what comes out, right? And not the 10 intermediate reactions. So what goes into glycolysis is glucose or another simple sugar. If you are a starch or something, you need to be broken down to a simple sugar before you can go into glycolysis, and that's not very difficult to do. So this is where all of your carbs start their breakdown, which is in glycolysis. You probably have memorized at some point in the past that cellular respiration occurs in the mitochondria, but not the first part of cellular respiration. The first part of cellular respiration, which is glycolysis, occurs in the cytosol. And fascinatingly, it actually predates mitochondrial existence. glycolysis existed before eukaryotes existed. And prokaryotes can do glycolysis, right? So this is a really, really old. They actually think that glycolysis predates high levels of oxygen on the planet. So,
the deep sea vents.
right, exactly. So glycolysis, you are still able to do glycolysis. Apparently it's been with us evolutionarily for billions of years and not just millions of years. So it doesn't require oxygen. It doesn't occur in the mitochondria. It occurs in the cytoplasm or cytosol. It's pretty inefficient, but it does get you a good intermediate product that you can use to get to some efficiency. Okay, so that occurs in the cytoplasm or cytosol, and it does not require oxygen. It can and does occur in anaerobic conditions. If you get into an anaerobic situation in which you don't have adequate oxygen, your only choice really is glycolysis. You can't do protein breakdown, you can't do fat breakdown, and you can't go any further with it, but you can send it right back through glycolysis. And we'll talk about that when we do muscle metabolism a little later this semester. Okay, so if oxygen is present, what's going to happen is the products from glycolysis you can send through the oxygen requiring parts of cellular respiration, which are Krebs and oxphos, and we'll talk about that in just a second. So if oxygen is not present and you just go through glycolysis once with a single molecule of glucose, you get a completely unimpressive level of ATP, but you do get a little bit, right? You get a little bit of ATP. So please notice in this figure that the things that you get are on this side and the things that you are required to put in are on this side, although they forgot to put oxygen right there. It's actually needed right there. Okay, so if oxygen is not present, you produce very little ATP, and lactic acid is formed as a byproduct, at least in some cells, and we'll talk about that with muscles as well. Not all cells can kick it back through glycolysis to produce more oxygen and generate lactic acid as a byproduct, but muscle can. Okay, so what happens is in order to get glycolysis going, it actually requires 2 ATPs to get it going, but it generates 4. So if we just keep track of the net, then of course you generate 2 ATPs from each molecule of glucose one time through glycolysis, right? Okay, so what we're going to do here is we're going to keep track, not of every single chemical reaction, but of where all of these reactants are used, which of the 3 stages, glycolysis, Krebs, and oxphos, and where the products are generated in which of the stages. Okay, you guys with me? So, in glycolysis, did we use glucose? Yeah, we did. Okay. Did we use oxygen? No, no oxygen. Did we generate any carbon dioxide? No, all the carbon dioxide is actually generated in the middle steps. Did we generate any metabolic water? Mm-mm. Did we generate any ATP? Yeah, we did. We generated 2 ATPs from glycolysis. So do you see what we're doing? We're just sort of keeping track when we go through each one of them. All right, so what do I generate as an output at the end of glycolysis? I get pyruvate, as I said, also called pyruvic acid. It's 2 of them, and they're both 3 carbon molecules, okay?
Are we keeping track of CO2 or does that?
We haven't gotten to CO2 yet. So CO2 is not generated until after glycolysis, and we will. We will keep track of it. Okay, so a biochemist would argue with me about this, but I'm trying to get you guys to understand physiology and not biochemistry. So what I'm going to do is lump this reaction with that one, because there's actually what's called the linking reaction that occurs right before Krebs. Okay, so this is called the linking reaction. What the linking reaction does is takes pyruvate and transforms it into acetyl-CoA. This is another coenzyme, acetyl-coenzyme A, okay? Acetyl-CoA is what is necessary to start Krebs, okay? All right, so I'm gonna lump all these together is what I'm saying, okay? This box goes here, these all go together, and then this box exists, okay? So, and then you're actually gonna go through Krebs. Krebs is 8 enzyme catalyzed reactions that you go through twice. What is the big deal about Krebs? Well, if you are a carb and you started up here, you are going to continue through Krebs if oxygen is available. Krebs occurs in the mitochondria. Everything that occurs in the mitochondria requires oxygen.
I put my phone on silent, so I don't know why I didn't turn on.
Okay.
Anybody else can turn their phone on silent?
Okay, so Krebs. If you are a fat, however, notice again that you skipped all of glycolysis, and a lot of times you start right before Krebs or right after. And if you're a protein, you start about these steps. Okay, so the reason I'm telling you that is not so much that I want you to memorize, but I don't want you to get into the habit of thinking that you only do catabolic processes on carbs, because you don't, right? You know, you can go a super high protein, low carb diet, right? Will you still generate ATP as a byproduct? Yeah, it'll be a little uncomfortable when you try to do it, but you will still generate ATP and maintain homeostasis. Okay, so Krebs uses pyruvate and converts it into acetyl coenzyme A. This requires oxygen, although I don't know why they didn't draw it in there. And notice what it generates as a byproduct. It generates carbon dioxide as a byproduct, and then it generates potentially some ATP, and only potentially. So let's go back here and figure out where Krebs is involved in the story. Does Krebs require any more actual glucose? No, it doesn't. Does it require oxygen? Yeah. Does it generate carbon dioxide? Yeah. Does it generate any metabolic water? No, metabolic water is all generated at oxphos. How about ATP? Well, with ATP, this is an oversimplification of kind of a complex process, but here's what I'm going to tell you. There are two different possible pathways for Krebs. One of them generates 0 ATP, and the other one generates 2 molecules of ATP. So what I say about Krebs is that it generates between 0 and 2.
What would be the determining factor?
It's what kind of molecule you're actually burning and under what circumstances. It's 2 different biochemical pathways that you can actually go down. So your textbook goes into excruciating detail about it if you want to know about it. Okay, so between 0 and 2. Okay, we'll just stick with that for right now. Okay, so now, who the hell cares? Now we still have not generated much ATP, right? 4 molecules from a single molecule of glucose. But the big deal is that this thing that you just generated at the bottom of Krebs is coenzyme 2H. It's another coenzyme, okay? And coenzyme 2H is what you need to get oxphos started. And oxidative phosphorylation is impressive because it generates 34 ATPs per glucose molecule. And that's where we were trying to get. All of this was just preamble, trying to get to oxphos. So if you were doing oxidative phosphorylation, some fatty acids start right before that. Most of the proteins start in Krebs. But this is where carbohydrates would continue to be broken down if you had oxygen, in the presence of adequate levels of oxygen. And it occurs in the mitochondria. And since it occurs in the mitochondria, it requires oxygen, exactly. And it produces metabolic water as a byproduct, and it produces 34 ATPs. So here's your metabolic water. So now we go back one more time. I know you know the answer. Do you use any more glucose? No. Do you use oxygen? Yes. Do you generate any carbon dioxide? No. You don't generate any more carbon dioxide. All of that comes from the middle. Okay. How about metabolic water? Yeah. Not that you really care, but it's good to keep track of it. Interestingly, in desert animals, conservation of this metabolic water is super, super efficient. That makes sense? It's one of the few places that they can actually, like little kangaroo rats in the desert, they have like super conservation of metabolic water. And then last but not least, ATP. How much ATP do I generate? 34. Okay, so now, from a single molecule of glucose, in the absence of oxygen, how many molecules of ATP do I generate? 2. Yes. Okay. From a single molecule of glucose in the presence of oxygen, how many ATPs do I generate? 36 to 38. Good? The others, I don't need you to keep track of numbers for any of the rest of it, but this is pretty damn important, right? Okay, so 36 to 38 ATPs per molecule of glucose for one time through in the presence of oxygen. Okay, and that's what you were trying to accomplish. Okay, so what are you going to need to know about this? What you're going to need to know is you should be able to write the unbalanced summary equation for cellular respiration, right? I don't need you to balance it. As a matter of fact, don't try unless you're really sure that you're going to do it properly, okay? I don't need to know about FAD or NAD or any of that business. We're going to keep track of the ATP, which is what we're going to be using, for instance, to run our sodium potassium pumps later this semester. So this is the relevant one that we're going to keep track of. Okay, so I need you to be able to do this. I need you to be able to do this, right? figure out where each thing is either used or generated. And then one last thing that you're going to have to think about, and that is that I need you to be able to discuss it. And the way that I want you to be able to discuss it is in 3 sentences or less. I want you to be able to tell me the relevance of cellular respiration to human physiology with specific examples. Okay? Which means I need you to be able to apply that. What do you think you're going to be able to apply it to? Any idea? See, what? Everything, right, but specific, right? So there's a whole bunch of different ways that you could apply, for instance, this.
Yeah, for muscle.
For muscle contraction, for any other, for catalyzing or powering any other anabolic reaction, for maintaining cellular homeostasis. Right, so what we're going to do is as we go toward the first test, which is where you're going to have to do this, you are going to see instances in which ATP is used. And you are going to try to put that back together with this story, which always seems completely not related and not relevant to anything else that you do in biology. What I'm trying to get you guys to do is to see the impact of this equation on, for instance, the sodium-potassium pump. Right, and what would happen if the cell didn't have a sodium-potassium pump? Lose homeostasis, cell death, right? So what I'm asking you to do is to log this in your brain to try to attach it to something in the near future, right? Before your first test. But it's going to be 3 sentences or fewer because I want you to be succinct. And what I'm trying to get you to do is simplify it so that it is a portable little nugget of knowledge about cellular respiration that you won't dump, right? Does that make sense? Okay, and so that's just sort of something that you're going to be thinking about and tossing over in your mind as we go through. And we will stop there for today, and we are going to finish this set of notes on Thursday, so make sure you bring the next one, okay?
Bring the next chapter.
Yes, please. Mm-hmm. I'm sorry that the quiz is